The human gut is the natural habitat for many vigorous bacteria, and we have yet to understand the physiology of many. Gut flora is one of the major bacterial communities found in the intestine, which is composed of around 300 to 500 different bacterial species with nearly 2 million genes. It can be found in both humans and animals, including insects. This bacteria can be established in many ways. At the beginning of the birth, the human gut is sterile, and once after birth, the bacteria colonizes in the infant's intestine through maternal birth, food, and other various contacts. Gut flora can lead to many pathological disorders such as colon cancer, inflammatory bowel disease, and diabetes. To begin, let's talk about diabetes. Diabetes is a type of metabolic disorder where patients are unable to regulate glucose metabolism, characterized by unnaturally high blood sugar, leading to a plethora of medical complications. There are two types of diabetes, which are referred to as type 1 and type 2 diabetes. Type 1 diabetes is due to the inability of the endocrine system to produce insulin due to an autoimmune response that attacks the beta cells of the pancreas. This type of diabetes is commonly found in children and adolescents. On the other hand, however, type 2 diabetes is not an autoimmune condition, but rather a condition characterized by somatic resistance to insulin, as well as abnormal insulin secretion. This type of diabetes is prevalent in adults. When most people think of diabetes, they think of an autoimmune disorder that's the result of diet or genetics, but recent research has shown that a number of factors play into the development of such diseases, including gut microbiota composition. Gut microbiota is a term that refers to all of the organisms inside the body's gastrointestinal tract. For humans, the 5 main phyla in the gastrointestinal tract include Actinobacteria, Bacteroidetes, Firmicutes, Proteobacteria, and Verrucomicrobia. The interactions between these bacteria, the human body, and diet have a large influence in the development of diabetes. In addition, they have also been implicated in the pathogenesis of obesity, chronic kidney disease, atherosclerosis, and even colorectal cancer, among others. The development of these diseases is related to the influence of gut microbiota composition on the human immune system. One example of this is how shifts in gut flora composition can cause immune disorders. In 2013, in a study done by Russell and others, vancomycin administration to young mice in early life development caused shifts in gut flora composition and enhanced future susceptibility to allergic asthma. These results were not replicated when vancomycin was administered to adult mice. In the study, Russell also explores a number of studies supporting the idea that microbiota composition during early life has a large impact on future immune system development. For diabetes, one explanation for gut microbiota-mediated pathogenesis involves the invention of antibiotics and the recent increase in vaccine use. Gut bacteria are not naturally exposed to most clinical antibiotics, since some strains of gut bacteria have a resistance to certain antibiotics while others do not. Antibiotics can select for an unnatural composition of gut flora. Similarly, vaccination may also lead to an artificial shift in gut composition. These shifts in composition may reduce beneficial gut flora, both promoting underdeveloped immunity and allowing for otherwise obstructed opportunistic pathogen growth. A compromised immune system, paired with an increased chance of opportunistic pathogen growth, results in higher susceptibility to a range of diseases, such as the ones previously discussed involved in metabolism, which can then lead to the development of diabetes. This condition of having maladaptive gut microbiota is called dysbiosis. Dysbiosis is one theorized pathway for type 2 diabetes caused by gut microbiota. In recent years, increasingly high consumption of highly processed foods has resulted in increased intake of carbohydrates and fats, along with significantly decreased fiber intake. Fiber, which cannot be broken down by the human body, is instead fermented by the gut flora, creating short-chain fatty acids, or SCFAs. These short-chain fatty acids produce immunoglobulin A and immunosuppressive cytokines, which cause anti-inflammatory effects. SCFAs also have an agonist effect on certain G protein-coupled receptors. G proteins, which are also known as guanine nucleotide binding proteins, are a type of protein that are involved in transmitting signals from a variety of stimuli outside the cell to inside the cell. Their functions are controlled by their ability to bind and hydrolyze guanosine GTP to GDP. G protein coupled receptors are cell surface receptors that receive messages in the form of peptides, lipids, sugars, and proteins. The binding of a signaling molecule to a G protein coupled receptor causes a conformational change, which then results in G protein activation and triggers the production of many different second messengers that help regulate a large amount of bodily functions. Activation of these G protein coupled receptors by short chain fatty acids causes secretion of glucagon-like peptide 1. This peptide impedes secretion of glucagon, possibly through antagonistic action of negative feedback loops. In addition, it hampers gluconeogenesis in the liver, improves insulin sensitivity, and augments satiety, all of which obstruct the pathogenesis of type 2 diabetes. Another theorized pathway involves bacterial secretion of butyric acid, which can boost the production of insulin. A recent study at Shanghai Jiaotong University compared the effects of a high-fiber diet versus that of a low-fat, low-carbohydrate diet on subjects with type 2 diabetes. The study found that out of those who consumed high-fiber foods, such as whole grains, 89% showed increased effective regulation of blood sugar, whereas only 50% of those who consumed the standard control diet did. The researchers theorized that this was a result of changing gut microbiota composition. In participants who adhered to the high-fiber diet, 47 bacterial strains were found to decrease in abundance, while 15 increased. These 15 strains secreted butyric acid. In addition, those who consumed more fiber lost weight and also had improved lipid densities as well. So in conclusion, the interactions of bacteria within the gut flora can lead to many immune diseases and disorders. Diabetes is one of the causes due to a compromised immune system caused from those bacteria. However, there are many ways to decrease the chance of type 2 diabetes, such as a proper fiber diet.